Oh my goodness. Yeah.
Isn't it interesting that that it's the flip side if you
Yeah.
right is left, left is
But
right.
on what they see, it's fine.
Yes.
But it can throw you. I know. Okay. So that. You're still muted. Good afternoon, everyone. Can you hear me? Thumbs up. We're getting so good at this. Welcome. Good afternoon, everyone. I'm just going to let somebody in. Oh, it's David Elkins. Look at that. Okay. Welcome to, this is our fourth, Dr. Brown? Fourth installment, and we'll be hearing Dr. Brown talk about the Nobel Prize for Medicine today. We're looking forward to that. And I would just like to thank David Elkins for last week. I can see him there. And I'm going to just, next week he's back and he'll be talking about the Peace Prize. And then the week after that, to close out the series, will be his wife, Val May, and she'll be talking about the Prize for Literature. So, you know, we have some great weeks ahead of us, and then we're already in discussions for what we're going to be doing in February. So sit back, relax, and enjoy. And I'm just going to get Dr. Brown going here. Give me 1 second.
So can you all, can you hear me? Okay, good. Well, we're starting off with Canadian content with this Paranthropus Robustness here, because this character was discovered by a Canadian anthropology student, a post-grad student in Victoria, BC, but she made the discovery in South Africa. And it's interesting because it illustrates the range in the appearances of our ancestors. Because look at the robust appearance of the jaw and the lower facial development here, which now that you're knowledgeable, you'll realize tells you something about what they ate. It was probably underground tubers and other hard to find and tough food. So it required large teeth, large muscles, probably a whole suite of genes would have changed and have caused this. But let's get on with this.
We have no sound.
Thinking about developing vaccines. And incidentally, 3 of them by Canadians or in Canada. So here we are, Harvey Alter and Michael Houghton. By the way, Houghton is in Edmonton, so he's moved to Canada. And Charles Rice, that was before the award. And it's, so this Nobel Prize was given for the discovery of hepatitis C, but there was a Nobel Prize given in 1976 for hepatitis, but in that case it was hepatitis B. So that gives you, and it was really into the 80s and 90s before this problem was run down. You know.
The sound is going in and out. We don't have sound now. Seems each time the slide changes, the sound goes out.
Maybe they don't want to see me. Ever think about that?
We can
So,
hear you again.
Well, now you have to see me. But anyway, the extraordinary thing is how much of a public health problem hepatitis still is. I mean, a lot of people die, and many more have chronic liver disease because of it, despite knowing what the virus is, despite having a vaccine, at least for hepatitis B, and despite having effective antiviral agents. And we've had them around for about 15 years. So quite a change. Now, let's go back before the 1960s or into the 1960s. Now, you know, I was thinking about this because I graduated in medicine in the early 1960s. I don't really remember this kind of number, but before that, roughly a third of patients who received repeated blood transfusions developed hepatitis. I mean, that's an extraordinary percentage if you think about it. And when they found the hepatitis B virus, that kind of cut the risk by about 20%, but it still left about 80% of them. So that's really what this story is about. And the hepatitis C turned out to be, its clinical behavior somewhat different than hepatitis B. Hepatitis A is more of an acute illness that most people get over, and it doesn't recur. But hepatitis B and C both have this propensity. There's an early, there are early acute symptoms that might last for a few weeks. And then this long, insidious period that might go on for several years, maybe several decades, before these patients develop cirrhosis of the liver, or in some cases, cancer of the liver. So it's a killer and a silent killer. Now, Halter, Houghton, and Rice characterized the cause of this hepatitis C and found that it was an RNA virus, just like COVID-19 is, an RNA virus. And so their pioneering work really led to not only the identification of the specific viral cause of the disease, but antibody tests for so that they could winnow out the bad blood from the safe stuff, and it also, I think, to some extent, led to the development of effective antiviral agents. But I think the last is much more true with COVID-19, or potentially truer for COVID-19 than is true for hepatitis C here. So, well, how big is the problem? Well, globally, about 70 million people have chronic hepatitis C. That's a lot of people. In 2016, almost 500,000 died from hepatitis C, from the chronic complications of the disease, meaning a badly scarred liver and or primary cancer, or both. And it's good news to know that antiviral agents cure more than 95% of those with hepatitis C. That's a real plus. But the failing, as I'll get to later, has been in the delivery of antivirals to those who need them, and to just finding out who's got it. I mean, it remains a serious problem in many parts of Africa, and certainly that includes Egypt and elsewhere in the world. Well, let's just go back a little bit on the history with the discovery of the Hepatitis B virus and talk about the risk of blood transfusions, particularly repeated blood transfusions, just increases the statistical risk that you're going to run into blood in those days that was carrying one of these viruses. But going back to that hepatitis B, sera from repeatedly transfused patients revealed an antigen, a kind of a protein signal, which was found to be part of a virus particle. And that led to the development of an effective immunological test for the virus. I mean, that's really important for deciding which blood is safe and which blood is not. And it also led, as I said earlier, to an effective vaccine. Now, there's that Nobel Prize. Baruch Blumberg received the Nobel Prize in 1976. Now, mind you, one of the three authors here, Alter, was a student of Bloomberg back in the 70s, and then moved to the NIH and took over the directorship of the blood bank. So he's really Alter's been in this business of hepatitis for several decades. By the way, all these three men know each other quite well. Well, what was the issue? Well, they thought they had the problem solved when they identified hepatitis B, the hepatitis B virus. But they only solved 20% of the problem, because 80% of the cases of clinical chronic hepatitis were happening, and it wasn't caused by hepatitis A or B, so-called non-A, non-B. Now, what did Alter show in all of this? Well, he showed exactly that non-B, that is, the cases that weren't caused by hepatitis A and B, was caused by some other virus. And he showed that taking serum from humans with non-A, non-B caused hepatitis in chimpanzees. So there was evidence that it was non-A, not A, not B, but something else with a big question mark. Now, they were able to, with electron microscopy, to show rather crudely that there was a putative virus, and it wasn't very big, 30 to 60, I think it was microns in size, but very tiny. Now, a little about the liver, because this is hepatitis, and if something happens to your liver, what are the symptoms from it? What does it actually cause? Now, this took me back because these two figures come from Grant's anatomy. I always liked, Grant was a professor of anatomy at the U of T, and for many years, all of these dissections, or most of the dissections, you could still see them in glass jars. And at U of T, I don't know whether he still can, but he was very good. He was an excellent teacher. And unlike most European anatomical texts that are cluttered with so many names overlapping the organs and what you're supposed to be looking at, it was hard to actually see what the illustrator was trying to show. Well, Grant didn't make that mistake. Well, the liver is a huge organ. Most of it's covered by your diaphragm. But if you kind of just reach under your right costal margin and take a deep breath, you can feel the lip of it just under your fingers. And that's also the region where your gallbladder is. So some of you who are missing your gallbladder aren't going to feel anything there. But the liver's rather unique because it has this portal venous system that drains the gut. I mean, from your stomach, the small bowel, and the large bowel. So that's carrying all the nutrients that have been absorbed and various other things might be toxins, and it ends up in the liver. And this disease, hepatitis, particularly if there's much scarring, or people with progressive heart failure get back pressure in this portal venous system, and it kind of backs up, and then fluid leaks out into the peritoneal cavity. And we're just going back to those functions. There's a production of bile, which facilitates the absorption of fats. It gets rid of that bilirubin. By the way, if you have liver disease, it's the bilirubin that causes the yellow eyes, the yellow sclera, and the yellow appearance of the skin, the accumulation of that. It's critically important for the metabolism of fats and proteins and carbohydrates. And in fact, patients who develop this hepatic encephalopathy or this brain disorder associated with severe liver disease, they're often made better by reducing the amount of protein that they're receiving. So it's also a storage place for glycogen, which is the source of high energy in the body, vitamins and minerals, and plasma proteins, and very importantly, clotting factors. So people with chronic liver disease often have a bleeding tendency. Now they also get into bleeding trouble because of that portal hypertension, because these people develop varices, like hemorrhoids, only their hemorrhoids are in the bottom end of the esophagus. And the trouble is, people can exsanguinate almost within half an hour or so if the varices are big enough and lose blood really quickly. So so that's the liver, and so some of the symptoms. One of the striking things in World War II, a lot of the service people who served in North Africa and the Middle East and in the Far East got hepatitis, although they didn't really know it was a viral illness then, but the striking thing was the fatigue and the nausea and vomiting, and of course the yellow eyes and skin. So the hepatic encephalopathy, that's an interesting one because I trained in neurology. I saw a lot of this. By the way, I should mention at this point, in North America, at least, and in Europe, the most common cause of liver failure is not hepatitis. It may be in much of the rest of the world, but it's alcohol for us. That's what causes chronic liver failure, at least 3 to 4 to 1. And almost all the patients that I saw with hepatic encephalopathy, it was from chronic alcohol. And that hepatic encephalopathy had a curious two distinctive features. One, the patients would simply go quiet, just quiet, not tight. They're not semi-conscious or unconscious. They're awake, but quiet. They're not interacting with their environment. And they developed what this was called this liver flap. You ask somebody to hold their hand back, like they just suddenly do that. It's all involuntary. So those are some of the clinical features. But look at these devastating numbers for hepatitis C, 70 million cases a year. This is in the era when at least you should be able to identify these cases and do something about it. And there we are, 400,000 deaths a year. Isn't that extraordinary? When you actually have the tools to do something about it. Or we have the tools. Now this graph shows that slow progression of cirrhosis. Cirrhosis is a fancy word for scarring in the liver. So this is with hepatitis C. So by, what is it, 20 years after exposure, so maybe when you traveled to Borneo or something like, or Indonesia or whatever it was, or South Asia or Africa or whatever, you might have had a little mild illness, didn't pay a lot of attention to it. And then 15 or 20 years later, maybe the first symptoms come up, or maybe annual blood tests reveal elevated liver enzymes, and you don't really have any symptoms. And so that 20% of those people at 20 years have evidence of scarring in the liver, and half of them by 30 years, and that continues on. So it's a really high risk, a terrible looking picture. Just take it for in the top right and the bottom. Anything that's blue isn't good. That's a collection of inflammatory, anything that's kind of red, reddish purpley, those are relatively normal cells. And but where there are holes and these blue strings is where the scarring is. And that's what the cirrhosis of the liver really does. It kind of compartmentalizes the liver. So you have these islands of relatively healthy cells, then these other areas, these partitions between them, somewhat impeding the blood flow going through them, and areas where cells have been destroyed, but largely where scar tissue has been laid down. Now, getting back to Alter. So he worked with this early Nobel laureate, and as I say, this about 70 or 80% of these cases that are non-B, there had to be another cause, probably a virus. And he showed that if you took blood from these patients that were non-A or non-B, you could produce this disease in chimpanzees, and they would develop the inflammatory changes in their liver and later the scarring. Now, Michael Houghton, this is our man in Edmonton. What was his role? He identified the virus based on genetic material from chimpanzees with non-A or non-B hepatitis, and also characterized it as, was able to characterize it as an RNA virus. Interestingly enough, in the view of, here we are with COVID-19, and Alter makes this point this year, after he won the Nobel Prize, looking back. He said it took 6 painstaking years to clone a tiny fraction of the hepatitis C viral genome. It took China less than a month to do it with this one and characterize the whole genome. So just imagine the extraordinary change. Well, we don't have to go through that, but one of the things, this is, you've all seen, you must have seen multiple pictures of COVID-19, what the virus looks like. Well, here's the hepatitis virus. Like all viruses, viruses can't make it on their own. They have to get inside cells and kind of take over the machinery, the molecular machinery of the cell to replicate themselves. So if you look at all viruses, this one's no different, hepatitis C, you have this kind of protein shell or glycoprotein shell. In this case, not spikes sticking widely out from it, but still a form of spike, proteins on the surface. And those are points of contact with cells that contact particular receptors, just the way the COVID-19 virus, those feet, those spikes, contact receptors on our cells. And there's the genome, in this case, an RNA genome like COVID-19. This one's a simpler one. COVID-19 has 30,000 base pairs. This one's roughly a third of that size. And it turned out one of the frustrations in actually proving that the putative virus, the hepatitis C virus, actually caused the disease was it was very difficult for them. That's what took up the 6 years, kind of assembling the bits and pieces, the Lego pieces of the putative virus, and then injecting it into chimpanzees to show that yes, indeed, this virus that we can, or they could map the genome in, actually causes the non-A, non-B hepatitis. And as it turned out, if you look at that strip at the bottom, the non-protein encoding end, when they tried that just by itself, and maybe a little bit more of the genome, the results were negative. They had to include other chunks of it, not all of that genome, but other chunks of that genome to actually develop a transmissible virus that, simulated hepatitis C. So now, I'm not going to get into this, but antiviral treatment is really very, very effective here. And so that's really good news. Now, one of the things that was interesting to me was that, you know, every time the Nobel Prizes come out in physics and chemistry and medicine, the Nature and Science have kind of review articles. And in this case, they went after, well, I mean, it points out a problem with the Nobel Prizes. All three of these men that I pointed out earlier, and like Jennifer Doudna, like 2 weeks ago, they led, in the case of the hepatitis story, fairly large teams. And it wasn't just one bright person at the top. They had postdoc students, very experienced people. And in this case, actually, these two people, Cho and Kuhl, I guess it is, I'm not sure I'm pronouncing that properly, were actually key to actually figuring out the virus and how to prove it, that it was in fact the virus. And there were conversations, I guess, in the lab. I mean, these people had some idea that this Nobel Prize is going to happen. It was deferred and deferred and deferred. And but they knew they were in the slot. They knew that they had been nominated, and that they were fairly high up the scale. And likewise, these two men knew it too. And the relationships were quite collegial, and they remain quite collegial as far as I can tell. But it is interesting that that Nature has this article, "The Unsung Heroes of Nobel-Winning Hepatitis Discovery." You know, how do you award things now? Sometimes, you know, the Nobel Prize nominating committee will nominate a whole group, but most of the time they're stuck with three, and they all have to be alive and kind of survive. Remember Penrose of 3 weeks ago, 89, and boy, you have to live long to make sure that you're going to snag the prize. And Houghton lost out because he died too early. So anyway, well, what are the remaining problems? I've alluded to them earlier. I mean, isn't it extraordinary that even now in 2020, with organizations like the UN and the World Health Organization, and that we don't seem to be able to get a handle on stuff like hepatitis. It remains a major killer, despite having the tools for identifying it and treating it. It's just extraordinary to me. And one of the problems is the high cost of treatment for those antiviral agents. Now, I think some people have some companies and families have contributed millions to that, but there's still hardly made a dent. Now let's get on to the COVID-19 stuff though. So we have the perspective of something, hepatitis, three viruses in what, 3 to 4 decades, kind of running down the specific viral causes, identifying them, and then developing a vaccine in the case of hepatitis B, and antiviral agents in the case of hepatitis C. Now, I remember writing something for the Lake Report way back in February or early March or something like this, kind of a theoretical one of what would we need, and among those two was certainly effective vaccine, and we'll talk about what that means in just a second, and antiviral agents. And by the way, there's been almost no conversation about antiviral agents, is there? Almost none. So I have to think they've been running into trouble in the development, because it's quite different than the vaccine programs. I mean, the extraordinary thing for me in looking at the vaccine programs is the incredible novelty and creativity in these vaccine programs. I mean, the way different groups in the developed world, and even to in a limited extent, the relatively underdeveloped world, Cuba has a vaccine program. Canada has 3. So all the way from using the shell of the virus, getting rid of the genome in the middle as the prompt, the antigen, to develop an immune response, using bits and pieces of the spike protein. The stuff that really turns my crank is the messenger RNA, or to a lesser extent DNA, to create the viral. By the way, messenger RNA, the 3 vaccines that have come out in the last 2 weeks, they're all messenger RNA. Remember messenger RNA? Messenger RNA is if you have the DNA in the nucleus of the cell, it has the template for all of the genes, but it by itself, it can't actually translate that code directly into proteins. It needs a go-between. Well labeled, isn't it? Messenger RNA. So what happens is that DNA, that double helix, kind of separates out. Then a single strand copy is made of that part of the DNA. That's the messenger RNA. And then that messenger RNA moves across the nuclear membrane and into the cytoplasm of the cell, and together with a structure called ribosomes, actually creates an assembly line for amino acids and proteins. So that's how it happens. So messenger RNA is really a code for making proteins. So if you can get a messenger, if you think about this theoretically, you don't know anything about companies, but you say, gee, I want to develop a vaccine, and I think those spike proteins are probably a really good target to go after. So I need a lot of those spike proteins around, but I don't want the virus around. So then you look at the part of the virus COVID-19 genome, that 30,000 bases, that actually codes only for one or more of the proteins in the spike protein. So that's all you're using, okay? You can't cause the viral infection itself. If you're Oxford, you use a virus to piggyback it into the cell. And if you're one of the other 2 companies, you piggyback it into kind of a fatty bubble into the cell. But all 3 are delivering a guide. Remember we were talking about Dna and gene editing? Well, this is a form of gene editing, isn't it? You're taking a selected piece of RNA, what Duda would have called a guide, the RNA guide, and you're injecting it in some way into the cell, either with a virus or not, and it creates copies of the target that you want, the protein target. The cell makes lots of copies of that, spills those copies out into the extracellular fluid in the circulation, where they contact immune cells in the bone marrow and lymph nodes. And some of those are B cells that create antibodies. And some of them are killer cells or cytotoxic cells that literally destroy on site any cells that are infected with the virus. So there you have it in a nutshell, how this actually happens. Now, if you use DNA, you're one step earlier, back up, because if you do use DNA to do this, it depends on the DNA, mind you, another selected strip of DNA to go into the cell to make the messenger RNA, and so on down the line. But that's the process for RNA and DNA. So we'll see a bit of it. Now, what are the goals of vaccine developers besides making a lot of money? And that's a hard knock. I should take that back. Although I will say this, you know, this some of these, the Washington Post had a really good article on this. In the early days of this, back before the warp speed stuff, Trump's warp speed program, developing vaccines is high risk business. In earlier times, unlike this, I mean, the riches of having the first three out of the gate, and they have an efficacy of 90% plus. Wow, that three of them, 3 out of 3. In earlier days, most vaccine programs failed, and any company that spent millions, or these days billions, trying to develop it might fail. So the venture capital been involved here. So, or government. And in the case of the United States and the European Union and Japan, governments have played a big part, and Canada to some extent too. Anyway, so you want something that has high efficacy. We'll talk about that in a bit. And that's the effectiveness in clinical trials. And then there's the real effectiveness once it hits the street and you actually find out the real numbers about how effective it is. And given the susceptibility of people my age and most of the faces I'm looking at, we want a vaccine that's effective in our age group. At least that's what I would like to see. And also you want one like the Oxford vaccine, and unlike the first two out of the gate, that travels well. You don't have to coddle it too much. You don't have to, you know, freeze it at -70 or -20. And it's kind of nice, like the Oxford vaccine, where it's low cost. It sounds like I think the Oxford vaccine is the winner right now. That's too early to say that, but I think it looks that way. And that's the way I thought about it early. So it travels well, no serious side effects. And one of the really big concerns with COVID-19 is that any vaccine hopefully lasts at least 2 years, longer than SARS or MERS, or the few weeks or a couple of months that we have with other coronaviruses like the common cold. So anyway, now look at these numbers, vaccine development phases, the preclinical. They haven't entered any clinical program yet. There are 190 out there, plus. Phase 1. Phase 1 is a very small trial, and all they're really interested in is trying to figure out what the appropriate dose is, and whether there are going to be any nasty surprises in a small group. Phase 2 gee, you get through phase 1, well, we need to look at this a little larger group, and that's phase 2, and also a little work on the dose. Phase 3 is when we get into what we've had with the 3 so far, 30 to 40,000 people. Roughly half of them not getting it, and half of them getting the vaccine. So you can understand that mounting programs like this is it's very challenging. It's one thing to be a molecular biologist and have these ingenious little ideas about messenger RNA and DNA. That's great, but running programs like this it's a tough business. And this is where the expense comes in, not messing around with messenger RNA. This is the stuff that costs hundreds of millions of dollars, is running a clinical program. Now, here's, I'm acknowledging this came from Washington Post. They did a great job just yesterday. And so I aped all of these, so thank you, Washington Post. So here's the coronavirus. It's that little ball with the spikes. You've seen this, right? And what is its target on all of our cells? This ACE2 receptor that we have in our lungs, we have it in the vascular system, we have it in our kidneys, we have it in the gut, we have it in our heart, we have it in the brain. So that explains the kind of multiple tissue cell targets in humans. So it's like a key in a latch. The key is the spike protein. The latch or the lock is the receptor. Click, it gets access. It's pretty simple structure. You can see that kind of wiggly thing in there. That's the messenger RNA. In this case, the whole 30,000 base pairs. And that messenger RNA takes over the cell's metabolic pathways to recreate itself thousands of times for a single cell. So much that the cell bursts and then spills all these thousands of copies out into the circulation. So that's the cartoon for it. Okay, here's the cartoon for the DNA and RNA vaccines. Actually, a good idea if it's, I don't know what the resolution is like for you here, but it'd be, I can't copy this out to everyone from the Washington Post. I can't do that, but this is so well done. Anyway, just pay attention to the bottom one, because we're looking at, on the bottom one, 2 of the 3 out of the gate first, okay? It's an RNA, so it's injectable, okay? And I told you earlier, it's a selected, it's a short piece of RNA that codes for 1 or more of the proteins on the spike. That's what it is. And it's enclosed in a little bubble of fat. It's inside the cell. You can see the cell of that big circle, and my finger's in the way. I can't point here. But anyway, you know, there's a butler here in the immune system. We call it the antigen presenting cell. So this is the one that greets the whatever the antigen is and says hi and introduces it. Its job is to shake the hand of the antigen and then introduce it to the rest of the immune system. And so that's in the little cartoon here. It introduces it to the B cells, which I alluded to earlier, bone marrow cells mostly, and they're the ones that create all these antibodies, these little Y things that kind of attach themselves and disable the virus. But it also sends a message to these T cells, T meaning from the thymus originally, not exclusively, but mostly. And one special, there are several little there are different types of T cells, but one of them is called a killer cell, and it literally lyses or destroys infected cells. So that's kind of a nice cartoon there. Now the three, look at these efficacy rates. Look, I mean, that's extraordinary. You know what the NIH in the United States, when they were starting to look at vaccines, their bar for success was anything over 50%. They would have been pretty happy if it was 60 to 70%. And look at what it is. It's 95, 95, 90. That's extraordinary. Now, these are early numbers. What's happening here? This is not somebody who's looked at 30,000 people in the trial. When you have a clinical trial running like this, you have a real time. They're following all of these cases in real time, and they're looking for the first statistical evidence of a significant difference. So what they're looking at is, the number that they're looking at is the number of people who develop clinical COVID-19 with a positive test. That's what they're looking for. Then they say, okay, how many of those got the vaccine and how many of the placebo? Follow what I'm saying? And so, for example, I've forgotten which one of them here, but I think it was Moderna, the one that's being done in conjunction with the NIH. There were, I think, I'm making this up. Let's, I don't have the actual figures. I think around 110 cases of COVID-19, and all but, I think, 7 or 8 of those cases were in the placebo group. And the remainder got the vaccine, but none of them who got a severe case. So it looked, and furthermore, it included kind of a representative sample early in the game of kind of older people at risk, which would be us. And so that's the kind, so you can understand why the various regulatory bodies want to look at more cases, not just the first 100 or 90 or 130 cases of COVID-19. They want to see a much larger number. In fact, they want to see as much of the 30,000 cases as possible. And so that's why, if it looks like foot dragging, it's very deliberate. And furthermore, if you're going to see an unusual but significant adverse side effect, you're more likely to see it if you look at data from thousands of cases, not just first 1,000 through the door. So, so that's how this stuff is done. Now, well, this is one to illustrate using a virus as a vector. This is something that's used in development of effective cancer therapies, gene editing, to use a virus that's kind of mute or that can't produce any disease, but it can behave as a vector to carry something from A to B. And in the talk that I gave on gene editing, it's used to carry a chunk of DNA or one particular, messenger RNA or the DNA patch for a particular gene into cells. And the virus, particularly viruses like HIV, not HIV, but viruses like that have that capacity to get into cells easily. So the Oxford group chose to use a viral vector to get their messenger RNA into the cells. So one of the concerns that was voiced about the Oxford vaccine early, before we had any results back, was, gee, this is an unnecessary thing to do, to use a virus to do this, because what would happen if the body developed an immune response to the vector? It essentially disables the vaccine. Well, it would seem that that didn't happen. Or you could vaccinate people with bits and pieces of the virus, bits and pieces of that spike protein. By the way, one of the Canadian trial group it's taking a kind of an interesting thing. All of these, or kind of 90% of these companies or more, have all made the decision that the spike protein is the most important protein to go after, which means that they're all, they may have different paths to the solution, but the target's the same. So if one fails, maybe the others will fail too. Or put another way, if you vaccinate a population and it turns out, gee, for the first few months, this vaccine really worked well, and then it didn't work so well after that. Well, what's the next option? Well, probably not to use something else that goes after that that spike protein. So one of the Canadian companies said, well, we're not going to go after the spike protein. We're going to go after some of the proteins on the shell itself, not on the spikes. That's a great idea, because we may end up in a situation here where the first vaccine does its job credibly well for a year or a year and a half or 2 years or whatever it is, and then the efficacy starts falling off. Well, what's the backup plan? Well, you better have a vaccine that's designed where the target is somewhat unique. So that's what, and oh, by the way, here's the traditional one. This is how vaccines were developed for years, right? You kill the virus or you stun it, you weaken it, and so that it can't produce the clinical disease, and you're essentially using the whole virus as the target. Now, I'm going to go through the 3 companies out of the gate. I think I mentioned some of this stuff, but Pfizer, that BioNTech, there was an interesting article and I think it was the New York Times, but I'm not sure about that. I have to look it up. On a husband and wife team, that's a small company. Only a year ago, they had 50 employees in Germany. And his wife is the CEO of the company. And he's kind of, I guess, the spark that kind of got it going. Anyway, they're an effective team. Now they're playing with the big boys here. And the big boys are Pfizer. That's where all the money is coming from, and certainly to run the clinical trials. And it was the first one out of the gate, but efficacy at 95%, and in those over 65, that's great. But this requirement to keep it frozen at -70. Now, how do you disperse that in communities or around the country or a community? That's a big hurdle. That's something that we don't have to worry about with the Oxford virus. Here's Moderna, which is also a small company, but they hooked up with the NIH. Fauci had a lot to do with this vaccine. All 3 of them are messenger RNA-1, efficacy at 95%. Look at that, no severe COVID cases in the vaccinated group. Now here's this 95 subjects developed a COVID-19, 90 in the placebo group, 5 in the vaccine group. Now, this one still has to be stored at -20, but that's a lot better than -70. And then the Oxford one, huge pluses. Efficacy is right up there. It included older age groups like us. It's cheap, like it's 10% or less of what those other two are, or projected to be. And standard storage and delivery requirements. So it would seem to have everything so far, but we'll see. Now, let's look at the world. 6 of them created by China, 6 by the US, 3 in Canada, UK 3, Japan 2, Australia 3, Germany 2, Russia 3. You know, Russia released that vaccine when they did a combined phase 1 and 2, but never did a phase 3, or haven't completed phase 3. And yet we're busily vaccinating people. India has 2, Cuba 1. That's interesting, and there are more countries I could mention. Now here are the 3 companies in Canada, and 2 of them are DNA vaccines. That's it. One of the second one down is interesting because here the DNA has been introduced into harmless bacteria, and there's no needle here. This you just swallow a pill, and containing these bacteria that contain the DNA, and then the DNA is released in the gut. It's a clever idea. I don't know how you quantitate a response like that. You kind of know what you've injected, but I don't know about that. But there are other delivery systems, and several oral vaccines in development. So here we are at the end. Now, isn't that an interesting contrast to compare? I'm not taking anything away from the hepatitis story. I mean, the obstacles were considerable. The technology simply hadn't evolved to where it has now, and but where there was maybe 1 or 2 companies trying to develop a vaccine, the kind of thing that only governments kind of have the money to take on. And look at this. And this is in less than a year. You think about it. The DNA, or the genome, was sent by email to around the world in early January. Well, we're now in November, late November. And by the way, that genome has been repeated many times. And why has it been done? Because is this a virus that's mutating on the run? And could our vaccines turn out to be useless in a year or 2? I mean, that's what happens with the common cold, isn't it? I mean, there's no vaccine for a reason. So all of that initial design work was taking place in January and February, and the stuff that's taken the biggest chunk of time has been recruiting big money into it, people that are willing to take the risk, or companies that are willing to take the risk, and then carrying out these clinical trials. But look at the number we've got compared to that kind of, what was it, 6 to 8 years for a vaccine for hepatitis C, and it was the only one. So this is a game changer. It wouldn't surprise me. I don't know what the Nobel Committee is going to do here, because who do you award anything to? But unless you give it to everyone that's been involved, and that probably makes the most sense, to acknowledge that, boy, science has really moved along big time here, and we're seeing the fruits of it. Now, we'll see if that holds up. Hopefully it'll hold up, and and we'll have several robust vaccines, and with some with different actions than others. And we can, if one doesn't prove to be effective for long enough, well, we have options. I think that's probably the way it's going to go. So we'll see. Now, next week, David Elkins is back talking about the Peace Prize. And then Valmay after that with the Literature Prize. We're really lucky to have those two. And then that's it for this year for the Nobel. So we'll see you next week with David. I can see David right there. And Valmay. Okay. All right. Well, goodbye. And we'll see you next week.
All right, everybody. Thank you for joining us again. We'll see you next week. If you have any questions, shoot me an email. I actually do have the link of the Washington Post article with all of those diagrams that you were
good.
showing. So I can
Excellent,
forward that
excellent.
to you people,
Very
and
well.
then you can look it up on your computer
Yeah,
on your own time.
one added thing I'll say. This is one of the other extraordinary things about this is the conventional medical literature. This has all happened so fast that the conventional medical literature just doesn't even pretend to keep up.
Mm.
Most of the papers on COVID-19 for months were unvetted and just out there to kind of look at. And that's what I found with this subject. It's just, there's a lot of really good information in public space. And maybe the New England Journal of Medicine will catch up in 6 months. Maybe. We'll see.
Well, for the time being, I'll send out the Washington Post.
Okay,
All right. All right. Have a great afternoon, everybody. See you later.
Bill, thank you,
Thanks,
Debbie.
Bill. Thanks,
Thank
Debbie.
you.
Okay. Yeah, that was fun. Yeah.
I'll just call Tim in and